everyone. It's very nice for me to be here today and with my old colleague, Dr. Bernhard, and also with Professor Aan. So welcome everyone to the 12th webinar of Indonesian Marine Biogeochemistry Forum. And today we will have two special speakers, Dr. Bernhard Meyer and Professor Aan Johan Wahyudi. And before we start, I invite Mrs. Idha to give opening remarks. Ibu Idha, the floor is yours.
Thank you very much, Bu Sulis. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. On behalf of Indonesia Marine Biogeochemistry Forum, I would like to warmly welcome all distinguished speakers, Dr. Bernhard Meyer, Prof. Aan, our moderator, Dr. Sulis, and all participants to the IMBF webinar series. Yeah, as Dr. Sulis already mentioned, today we have two honored distinguished speakers. The first speaker will be Dr. Bernhard Meyer, who will share insight on emerging technologies in marine biogeochemistry. Once again, I would like to say thank you very much for your willingness to share with us today, Dr. Bernhard. It's truly an honor for us. And the second speaker will be Prof. Aan, our chairman. I will highlight marine biogeochemistry research in Indonesia. I believe that today's discussion will spark valuable insight and strengthen our collective commitment through this forum to advancing the marine biogeochemistry research in Indonesia and even beyond. So before we begin, allow me to share two announcements from the IMBF organizing committee. So, Sulis, may I share my screen?
Yes.
Yes. So don't worry, everyone. It's just one slide of PowerPoint. I don't want to talk much today. I want to hear Prof. Bernhard and Prof. Aan. So the first announcement is about the training. So again, IMBF will held a training for the scientific writing training, and we training on 2020 with, of course, featuring distinguished trainer. And I'm sorry, it's only for limited participants, but this will be free of charge for the IBF members. So please anticipate the announcement in the group, and I expect to have some applicants apply to us. And the second announcement is about the IMBF webinar series. We successfully completing 12 inspiring sessions featuring distinguished speakers. Not yet. Today is the 12th, so almost 12. And we will have webinar season 2 next year, start from January 2026. So we invite all of members to contribute by sharing ideas. Of course, Bernhard, you can share if you have any ideas or topics that will enlarge our engagement network. We will happy to hear that. For the our upcoming webinars, please contact us at the platform, either in email or our social media. Yeah, fine. I would like to express my sincere appreciation to our speakers, Dr. Ben, Prof. Aan, our moderator, Bu Sulis, and all participants today. I wish you have a fruitful and enjoyable webinar. Thank you very much.
Thank you, Ibu Idha. Okay, I think we can move forward to our next agenda. I'll invite Dr. Bernhard Meyer to give us the presentation entitled, wait, the title is this. Okay, the title is Measurements, Lab Experiments, and Numerical Modeling to Investigate the Fate of TBOC on the Sunda Shelf, Southeast Asia. So the time is 20 minutes. Dr. Bernhard, the floor is yours.
And I share my presentation with you, if I may.
Yes, can you share your screen?
I try.
Okay.
Does it work?
Yes. Oh, yeah, we can see your screen.
Okay, and can you see my mouse also?
Yes, we can see it.
Very good. Okay, so then I can start, yeah?
Yes.
So this is, I would like to present some results from our project, which combines measurements, lab experiments, and numerical modeling. And the investigation topic was the fate of TDOC on the Sunda Shelf in South. So TDOC, I have to say also that there were some scientists involved in this because it was really a broad project and much data. This is why we have so many co-authors and so many institutions joining this and helping, supporting, which was a very nice project. So what is TDOC? TDOC is terrestrial dissolved organic carbon, and this is transported essentially rivers from land to the ocean, to the coastal ocean. And the TDOC is undergoing remineralization. It's degraded, and it will change from the DOC, dissolved organic carbon pool, to the dissolved inorganic pool, where a compartment of the CO2, which influences the pH value, its outgassing, and so on. And so TDOC is very important because it is remineralized and it changes the coastal biogeochemistry system. TDOC is furthermore very, very complex because it's undergoing these degradation processes. And we have the pure biodegradation made by microbes, which are kind of eating the DOC and producing DIC again. And we have photodegradation, which is coming from the radiation, essentially from the UV radiation, which is also degrading the DOC. And we have an interactive photobiodegradation. And this is very often underestimated. And the degradation rates are not yet understood actually and measured. And this is why the TDOC processes really modeled yet so far. We are starting to do this. Where does it come from? I said already from land, of course. So this is a map showing the peatlands in Southeast Asia and India essentially, and Malaysia. And this is the main, the peatland, of which we have some river draining the water from peatland into the coastal ocean. And the contribution in Southeast Asia from this map is about 10% of the global flux of organic carbon from land into the ocean. So it is an important thing to investigate because, as I said, it changes the biogeochemistry in the ocean. So our motivation about this was how much TDOC do we have in Southeast Asia? How much is the input into the Sunda Shelf? This was not yet really fully understood as well. How much of this coming into the Sunda Shelf is degraded on this shelf, and how much of it is exported directly or indirectly into the so-called world ocean, which means here Indian Ocean, and then transported further maybe to the Atlantic Ocean and so on. And how does it change the shelf carbonate system? So yeah, this is what we were aiming to. So let's start with the field measurements. And this was about the riverine TDOC input into the coastal ocean. For this, we could use older field measurements, actually, which were done during the SPICE cooperation between Germany and Indonesia. And what we used were carbonate system parameters in river mouths. They have been measured like alkalinity, total alkalinity, dissolved inorganic carbon. From these two, we can estimate all other parts of the carbonate system, like pH value, for example, and so on. Yes, and what we also need to estimate the amount of parameter input into the coastal ocean is a map of the peat coverage, because we found that there is a relation between the parameter concentration in river mouths and the peat coverage of the catchment area of that particular river. And this map shows the peatland distribution, and we could overlay this with the catchment area map of our hydrological model. And so we had the percentage of the peat cover in the catchment area, and from this we could estimate the concentration and, at the end, the input of mass of the parameters into the coastal ocean. And what we had then, this shows measurements depending on the peat coverage percentage of certain rivers. In this case, pH value was measured, and PCO2, the partial pressure of CO2, was measured. And depending on the on the peat coverage in percent, we have this graph, and from these distributions we could these equations displayed by the black line could then be used in the model. So for each parameter, we could calculate, depending on the peat coverage, the concentration, and with this, the input of this parameter into the coastal ocean. For the CDOM, in this case, 380 nanometer, we found that it is a function of DOC concentration, and it looks like this. And this was also something we could use in the model. So let's come to the lab experiments, and this was about the degradation process. This is also a new setting, what was used by the group of Patrick Martin in NTU, Singapore. Because, as we said before, interactive photobiodegradation is very important, and so far it has always been measured separately, the photodegradation itself and the biodegradation itself. And here, the setting of the experiment combined both. So we had a chamber with constant temperature with a TDOC sample, and this was pumped through a quartz tube, which was then illuminated, and we knew exactly how much illumination, how much radiation the samples got. In this case, we could also measure the photodegradation, and the outcome was something like this. So this shows the TDO concentration. The x-axis is the time. And once in a while, every week, for example, samples were taken out. There was a dark experiment without any light. And this was this curve, the degradation rate, as we can see. And the light exposed treatment produced this curve as an example only. Sorry. And from this, we could calculate all three types of degradation. And from this, we could also create equations again, which could be then included into the model. So we have pure biodegradation, the interactive photobiodegradation, and we have pure photodegradation. And this is what we put into the model as well. And the result actually was something like this. It's an exponential degradation rate for DOC depending on the UV absorption, which was unified by the DOC concentration. And this was integrated into the ecosystem model. So I will come to the modeling. The model was about the degradation and the transport of TDOC, the input as well, of course. And from this, we could estimate the export from the Sunda Shelf and the effect on the carbonate system. The model system I would like to present briefly. This is the model domain. And these red dots and violet dots are river input points. It was altogether 1,700 river input points in our domain, and the violet were the peatland, where we have more than 10% peatland in the catchment areas. We have open boundaries along the Indian Ocean and the Pacific Ocean and South China Sea and so on. And so we had to apply first a global model delivering information along these open boundaries. So our model setting was the following. We first applied this MPIOM, which is a general circulation, global circulation model. We applied also a hydrological model and HydroPy to get the river discharge of fresh water. The next step was to apply the regional circulation model HAMSOM, and this was about producing realistic circulation and realistic distribution of temperature and salinity. And after that, we subsequently applied the EcoHAM, which is an ecosystem model to simulate the regional biogeochemistry. And here we put the river discharge and the input of the biogeochemistry parameters in, as well as along the open boundaries here. And this model then could simulate the advection across the domain and the TDOC degradation, and with this, the influence on the biogeochemistry of TDOC degradation. And we were running 2 simulations for the last 60 years approximately, one with TDOC degradation to the DIC pool, as I mentioned before, and one simulation without TDOC degradation to see the effect of this degradation. This shows, just for verification of the quality of the ocean circulation model, a comparison of measurements in the Singapore Strait of salinity, sea surface salinity and sea surface temperature in red, compared with the model results of HAMSOM. And we see very nicely that the seasonality is met and the values themselves are also very, very good in agreement between model and observations. And the same with biogeochemistry parameters like total alkalinity shows also the seasonality, the DIC, dissolved inorganic carbon, and the DOC as well. We see here, this is what I would like to point out also, during the summer monsoon season, the south monsoon season, a flow which is coming rather from the Malacca Strait into the Singapore Strait, so eastward transport. And during the winter monsoon season, we have a westward transport more in the Singapore Strait, where South China and Pacific Ocean water is transported into the Singapore Strait. For the PCO2, the partial pressure of CO2 in the water and the pH value, this is all also very nicely in agreement with observational data. Observational data are always black lines. All right, let's come to some results. I would like to show how much is put into the South China, into the Sunda Shelf seas, and where does it go? So the inner Sunda shelf is defined in this case by these thick red lines. And if we look how much is input and how much is degraded, we see that 51% of the input coming through the rivers onto the Sunda shelf is degraded on the Sunda shelf already. So outgassed, I said already that the DOC is going to the DIC pool, which means increase of CO2 concentration. And this increases the outgassing of the water, of the CO2 from the ocean water on the Sunda shelf. And the 20% of the input, which is coming through the rivers onto the Sunda shelf, is therefore outgassed into the atmosphere. And how much is exported? 28% of the input through the rivers is exported directly to the Indian Ocean through the Andaman Sea or through the Sunda Shelf. And 20% of the input is exported through the South China Sea or the the Java Sea, the Java Sea, Flores Sea. This going out the South China Sea may be recirculated, but this is not taken into account in this budget. Horizontal map, the effect of DOC degradation. And for this, I would like to focus on the third column, which shows the difference between the simulations with minus without degradation. And the first map shows the pH value, where we see that it's decreased, of course, because the CO2 is increased. And it's partly even decreased up to 0.05 units as an average for the upper 50 meter during the last decade of the simulation period. The CO2 flux is increased, as I said before, by 2.3 units approximately. So we have more CO2 flux from the ocean to the atmosphere. And the omega aragonite, this saturation state of aragonite, which is very important for the growth of corals, for example, is also decreased, of course, because we increase the CO2 inside the marine. Yes, and what we see also, which is clear, that the largest effects are always close to peatland. And since the Malacca Strait is very much affected by peatland rivers, we see always the largest differences in the Malacca Strait. Last but not least, some time series. This is a time series for the last 10 years again of the simulation until end of 2023. And here we see the trend also and the difference between with and without degradation. So with is in red shown and without is in green shown in green color. And what we see is that the PCO2 is increased by up to 30 ppm in the water. And here we see also that the trend is much larger if we have degradation than if we have no degradation. The pH value is decreased, as seen before, on the average by 0.02 units, which is an acidification, of course, of the water. And the aragonite is decreased by 0.1 on the average. And the trend is also much larger if we have degradation. So the raising TDOC input is followed always by raising TDOC degradation, and this increases the CO2 concentration in the water. And if we change the land use and dry out peatland, we increase the flux of TDOC from the land to the ocean, which ends up like CO2 emissions to the atmosphere, like industrial countries. from the industry. All right, some conclusions. So we were using a new approach for the interactive photobiodegradation to measure the complete degradation rate from these three different types of degradation. With this, we found a new parameterization for the TDOC degradation in the ecosystem model, and we used this, and we showed that realistic long-term circulation biogeochemistry simulations are possible with this setting. And the next step would be, or should be, and I hope will be, the climate change simulations to see what it will look like in 2100, for example. All right. This was my contribution so far, and I'm happy to be able to present this. And yeah, I'm open for questions or later. Thank you.
Yeah, thank you very much, Dr. Bernhard Meyer. Okay, so we will move to our next speaker, Prof. Dr. Aan Johan Wahyudi, the chairman of the IMBF. The title is Marine Biogeochemistry in Indonesia, Research and Future Direction. So, Prof. Aan, the floor is yours.
All right, thank you very much. Yeah, following up for the previous presentation by Bernhard, I would like to present my colleagues and I that study about the progress of biogeochemistry research in Indonesia. And I changed a little bit the title, past lesson and future direction. And this is the team that involved in this study, including me and my colleagues in Research Center for Oceanology of National Research and Innovation Agency. As the background of the study is that we have the marine biogeochemistry in Indonesia, which have the key insight, which including the control cycling of CNP and silicon regulate that regulate productivity and also ecosystem resilience and also climate, including the carbon dioxide exchange and carbon sequestration. Of course, the carbon dioxide exchange means the carbonate system and also the piece of organic or piece of inorganic carbon, like previously mentioned by Johan. And also the marine biogeochemistry also include the myocarbon and nutrient sink or source, which is then regulate the pH, including burial, remineralization, and also affecting the ecosystem health. And now currently we have the urgent challenge, including the ocean acidification, which also the previously mentioned by Bernhard regarding the study in Sunda Self. And the other challenge including dioxygenation, eutrophication, and under the climate change. Now we have limited in situ data. Of course, we have sensor-based data collected from many joint crews or joint research project, but also we actually still have limited in situ data since the most recent bias to temperate region and lack in the integrated synthesis of especially in the locus of in Indonesia seawater. And yeah, if we talk about the marine biogeochemistry research in Indonesia, is still fragmented in data and bias toward temperate region. And now we think critical knowledge gap in understanding how socio-environmental change affect ocean cycle and management strategies. So we aim to synthesize marine biogeochemistry research in Indonesia, especially in the period of 1950 until 2024, which revealing trend, gaps, and contribution while strengthening its global scientific and policy relevance. With the objectives including access advancement and knowledge gap in key areas, carbon cycling, nutrient hypoxia, pollution, and climate impact. And the second is to highlight methodological improvement and future need for ocean observation system and propose priority biogeochemical parameters for conservation and sustainable management strategies. And the scope of and data acquisition, including the marine biochemistry research conducted especially in Indonesia and its surrounding water, which mean including the Sunda South Sea, which is not only belong to the administratively belong to the Indonesia, but also the surrounding countries, such as Malaysia, Singapore, Brunei, etc. And we also assess for example, the South China Seas or the Eastern Indian Ocean or Western Pacific, which facing the Indonesian maritime continent. And the key themes that we collect from the bibliographic data is marine biogeochemistry, including nutrient dynamic, carbon cycling, and pollution. And the bibliographic data were retrieved from Scopus, Crossref, and also Google Scholar. We use Google Scholar as complementary, since the Google Scholar will able to collect the gray document, including the national journal or report that not published in international journal. So we hope that using this kind of source of the bibliographic data from three well-known source, we can objectively cover all of the marine biogeochemistry studies conducted in Indonesia. And we use the search focus on the key research area, using several keywords or keyword combination within the key themes that previously mentioned. And the method we employ is that systematic literature review using conducted to comprehensively assess the marine biogeochemistry research. And the approach is the preferred reporting item for systematic review and meta-analysis, and well known as PRISMA. And we collect in this chart that we firstly record the identified database, which is about 15,000 more documents. And then from this record, we selected and remove those that duplicate or record that record marked as ineligible, and record that removed for other reason. But in the first stage, we remove the duplicate record and we get 11,000 and more. And then from this record, we exclude due to the irrelevant topic or theme of the studies. For example, that those studies that not within the Indonesia or surrounding water. And then we finally got, after the assessing for the eligibility, including those that excluded because of irrelevant and then freshwater ecosystem or wrong study design, means not eligible for the biogeochemistry research, and those research that not belong to the the maintain, which is, for example, the policy or management and terrestrial ecosystem and etc. Then we finally create the 472 studies the special marine biodiversity tracking. And the first is that bibliometric analysis to know the past studies trend, including network visualization, clustering, interconnected research using the course viewer tools and Python code pipeline. And we analyze the future projection based on those 472 documents that were categorized into thematic domain, employing a keyword-based tagging system and using the tools of artificial intelligence, ChatGPT 4.0.0 and Python code pipeline. And the progress of marine biogeochemistry research in Indonesia can be divided up to 3 stages. The first is early development in marine biogeochemistry studies, which is a flashback to the beginning of 20th century until 1950, and then the growth and diversification, which start in 1950 until the beginning of new millennia. And the third is the 21st century advancement, which is the past 24 years. As for the early development of marine biogeochemistry studies, including the foundation in pre-19 until 1904, which is we recorded the early expedition such as Challenger and Sibuga expedition, which involved the foreign scientists, mostly from England and Netherlands, which laid the groundwork for understanding oceanographic and biological variability, and also the establishment of fisheries station in Jakarta, which is 1904, and that institutionalized the marine science in Indonesia. And then the pioneering research, which is up to 1930s, focused on fish larvae, which is mainly on marine biology, but it's become the foundation of the marine biogeochemistry research as well. That focus on fish larvae, coral reef, marine algae, with key contribution by very well-known scientists at the time, including Detzman, Firby, Heidenberg, Weber, Van Busse, and including the major expedition like Senelius expedition and the Galatea expedition, that advanced knowledge of nutrient productivity and the deep sea of Indonesia. And start in 1950s, that the transition to local leadership, which is the growth of Indonesian expertise and institution, including the Academy of Biology, previously in the Indonesian Institute of Sciences, that marked a shift toward national leadership in firstly in marine biology, but then it's become the foundation for marine biochemistry research in Indonesia. And during the growth and diversification, we can divide sub states, which first is post-independent foundation, and then the research and international cooperation start in 1970s, and then systematization and ecosystem level focus, which is in the late of 20th century. As per the 21st century advancement, which is start in 2011, and then the moment we get the data from the document we have, which is 472 documents, including journal proceeding report, that there are a significant increase in marine biodiversity research after 2000. And the trend is accelerated in 2010, which likely driven by advanced satellite remote sensing and biogeochemical modeling, a rising interest in ocean acidification, climate change, and eutrophication, which is the main challenge of our ocean, and Indonesia participation in global marine science program such as GOOS, Global Ocean Observation System, and integrated marine biosphere research. And also the expansion of marine biogeochemistry monitoring network that involve not only the national in Indonesia, but also the regional in Southeast Asia, even through the Intergovernmental Oceanography Commission, Western Pacific Commission, and also the Indian Ocean Research Program. And the primary cluster of the past marine biogeochemistry research can be divided into four main clusters based on the bibliometric analysis, which is the marine and ocean coastal contamination and heavy metal pollution, which is basically marine pollution, marine sediment dynamic and water quality, and carbonate system and organic carbon cycle. This is four clusters according to this analysis, which is green, the cluster the color green, then red, blue, and yellow. And we also analyze how that primary cluster evolve in per year from the beginning until the recent years. And we know that for the cluster number 1, which is main productivity and ocean biochemical process, basically start before 2000 and then still ongoing until this recent year. And then the cluster number 2 is that sediment and also and pollution is also evolving until the recent year. And this cluster illustrates the diversification and deepening of marine biochemistry research, which is transitioning from isolated studies toward the interdisciplinary, that problem-oriented investigation that address both ecological function and environmental change. So this is how each cluster evolve year by year. Of course, we can see here for the TIC in organic carbon, this cluster number 4, carbonate system and organic carbon cycling is not well studied in the past year, so will be good sign. For example, this, like previously mentioned by Bernhard regarding the carbonate system with the, for example, Indonesia or other Indonesia water. As for the future projection of marine biochemistry research, we use thematic keyword dictionary were constructed comprising 5 core research themes relevant to future marine biochemistry research in Indonesia. So the 5 core research theme is represented Indonesia Ocean Research Agenda, with we call it Agenda Research Samudra, which previously disseminated through the Ocean Research Consortium and Indonesia Marine Research Foresight by Ariana et al. In 2017. And we did the cross evaluation of marine biochemistry research theme across 4 dominant clusters. Previously, we had 4 clusters of dominant previous research, and also the 5-10 that potentially for the future direction. So we, after cross evaluate these 4 cluster and 5 main themes, before the use of this approach is to establish the baseline research focus for the future projection. The distribution of 472 studies across major biochemical theme and cluster that were examined. And this analysis revealed that anthropogenic pollution and carbon cycle themes dominate the landscape, where observing system and climate feedback are notably underrepresented. So we have 5 research themes, which is climate change feedback and ocean observing system and ocean carbon cycle, pollution and anthropogenic stressor, and regional policy collaboration. And the 5 core research themes, which is we know that if we flashback for the past lesson, the temporal trend of major marine biochemistry research theme previously showing increasing focus on ocean acidification and policy-related studies, while the pollution studies are declining, unfortunately, and maybe due to the study that already goes through so many studies about the pollution, so it's declining because of the interest is probably also declining. And the strategic implication of that kind of the trend is that we use the quadrant-based gap trend analysis that conducted by comparing historical coverage with recent publication growth, that allowing its trend to be placed within strategic typology, highlighting the areas of strength, saturation, emergent, and neglected. And we can see here that we have 4 quadrant 1, 2, 3, and 4. And we previously I said that pollution research with comprehensive biochemical of the pollutant is declining in the trend because of the historical coverage is high, but the trend of score to the recent or early studies is lower, and shows that these studies is saturated. And the other studies like ocean carbon cycle or policy collaboration emerging, but the climate change feedback and observing system is not mature yet, so it still need the progression. For the explanation is that quadrant 1, which is highly mature and fast-growing theme, like the ocean cycle, and which requires support and integration with advanced modeling and say. And the quadrant 2, which is cover emerging priorities. And previously, in this figure, that including climate chemical feedback and marine feedback impact, and low current coverage but high growth, which is warranting early investment. If we do the investment in this area, then we hope the progression is ongoing smoothly. And the quadrant 3 with the low coverage and flat trend, that marks knowledge gap in area like ocean observing system, suggesting a need for capacity building and investment. And the quadrant 4, which is into saturated and established field, such as pollution research, marine pollution research, where efforts should shift from baseline mapping, which is observed field observation or field research to integration and mitigation, and also for policy making. And this is how the future research priorities in marine biochemistry. And we have 5 priority areas, and the key focus of each area and coverage and trend on the past 20 years, and then gap status. Basically, the climate change and biochemical feedback is emerging priority. Ocean acidification and carbon chemistry high priority. Pollution anthropogenic structure is saturated and declining, but coverage is very high, so we have to move to the next step, which is to mitigate and also to employ the result of the pollution research to the policy making. And also the technology innovation on observation, which is including the ocean observation system, coverage is very low and the trend is flat, so it's critical gap. So that for the next future, we have stick to through of this kind of research. Since we now have the autonomous platform, AI-driven modeling and alert integration, etc., which essentially will make this trend growing progressively. And also for the policy and regional collaboration, it's also emerging priority. So if we also see how the pollution and anthropogenic stressor is already saturated, we can combine this kind of research for policy and regional collaboration, so that the research will still growing and emerging. As the take-home message is that strong growth in marine biochemistry research in 2000, but of course still fragmented and coastal focus, and major gap is ocean climate interaction, carbon budget, long-term monitoring and modeling. And since the observational capacity limited by weak time series, scarce autonomous platform, and data integration gap, so in the future we need to pay more attention in this observational capacity. And of course, we understand the lack of a national program and the reliance on short-term projects that hinder continuity and policy uptake. So we need to better strategies that mostly of the program
Ya anjing, udah nggak boleh pakai LinkedIn anjing.
is the due to the project based program that with a short term or limited time. So we need to more strategies to overcome this limitation. And future progress depend on sustained observing system, of course, also early career support, science policy integration for SDGs and climate action. That's all from my side, and thank you very much for your attention.
Yeah, thank you very much, Prof. Aan, for really interesting presentation. We still have time, so I will open the next session will be the question and answer. So I will open the first 3 questions from the audience. Is there any question? You can write on the chat box or unmute yourself and give the question.
I have a question
Yes.
to Prof. Aan. You said there is a gap of observational data, right? I have the feeling that, like in many other countries as well, there are how to say, lots of observational data which are not really published. I mean, they could be used and analyzed. They have been collected during research cruises, for example. I know that the former LIPI, for example, they performed many cruises and collected lots of ocean data, which may not have been analyzed entirely because it was too much, for example. So are you planning or thinking about asking the former institutions to make data available? This is, I think, a point which could be helpful.
Yeah, so can I respond?
Yes,
Yeah,
Prof.
so yes, yeah, we aware that from previous joint research or joint cruise, we have already collected in situ data from the joint cruise or also the sensors that deploy in the ocean of Indonesia water. And yeah, we aware of that data availability. As you mentioned that the data is scarce and in the respective institution at that time, and now we start to collect that data and not finished yet, but it's progressing. Since, for example, we have the national repository for the data, and I see that the data that collected from previous studies on the previous cruise, some of them is already there. So basically, it's there are limitation of the access, but we can ask for the owner of the data in that kind of platform to get the access of the data. And the other data is basically open access data that we can find. And so with this kind of platform, we can use the data for example, validating the model or validating the satellite-based data set. And also, this year I read preprint paper of the Indonesian college, especially physical oceanographer who collect all of the data available in the past 2 decades of Indonesia, especially in physical oceanography data, and collect them, and then in process of publication in the data science journal, that we can use, of course, this kind of data set for future collaboration and also future research as well.
Yeah, thank you, Prof. Aan. Is there any question from the floor, from other audience? Not yet. You could also write the question on the chat box. Okay. So while we waiting the question from other participant, I have one question for Dr. Bernhard Meyer. Is this regarding the refer input? I was wondering what kind of, the source of the input data that you use for the models. Can you explain more about that? Because I know that it's quite difficult to find data set for refer input, such as for the differ discharge and also for the nutrient input from the river.
Yes, you're right. It's a challenge to get information about the input. But as I mentioned before, we used majorly actually the SPICE observations, SPICE measurements, and related the concentrations which were collected, which were measured actually at river mouth to the percentage of the peatland in the catchment area of that specific river. And with this, we could create really equations which were I showed this, I think, which were, shall I show it again?
Yes, You can search again.
So what I talk about is this one here. And here we can see the red dots are measurements, like for pH value, for example, and PCO2. These are two parameters amongst others which have been measured at river mouths. And we know the percentage of peatland in the catchment area of that specific river. So these were some different rivers in Indonesia and Malaysia, I think. And from this, we could really create these curves. And this is what so these curves say, give an information about the concentration of the particular parameter depending on the peat coverage. And since we had the peat coverage for each river, input river in our model domain, which was calculated by overlaying the catchment areas of each river from the hydrological model with the peatland distribution. From this, you can calculate the peat coverage in each catchment area. And then you just have to apply these equations. And with this, we calculated the input, the mass input at the end of the different parameters of the biogeochemistry system, and the TDOC and CDOM as well.
Okay. So do you also include the seasonal variation also in this input in the order for input?
Sorry, I didn't understand. If we considered what?
The seasonal variation.
The system.
The seasonal, like different season. Yeah.
Yes, we did, because we had the what we used. We calculated the river load, and the river load depends on. The river load is a mass of a certain parameter transported into the ocean, into the coastal ocean, and this depends on the river discharge.
Okay.
Yes, and for the river discharge, we had daily values. And these daily values for the river discharge of water was created by the hydrological model.
Okay. Okay. Thank you very much, Dr. Bernhard. So is there any question from our audience? I'm still waiting. We still have time until 15:30. Don't be shy. Please ask your question. Okay. Not yet. I'm still waiting. Okay. So maybe I have one question for Prof. Aan. I think you mentioned that we see that, I mean, in general, the observing system, especially for biogeochemistry parameter, is still is very lack. Maybe do you have maybe any idea or insight how can we improve this in the future, especially in Indonesia?
Yeah. So the main challenge at the moment is that the observing system that include the biogeochemical parameters is considered expensive or costly, especially in Indonesia. And it's different from the physical oceanography parameters that we rely on the sensors or in situ observation using the sensor or like mooring buoy or other sensors. And for the biogeochemistry variable, we need to collect the sample, sometimes using sensors out there, and also the bottle sample that should be bring to the laboratory for further analysis, etc. So this kind of collecting data or data acquisition is considered costly, and that's why it's become the main challenge at the moment. But for the future progression, I think that we need to start um, um, especially actually we already have the, like Ben had previously said, that the previous joint collaboration, joint cruise and extra, we already have the in situ data, including the biogeochemistry parameter that actually we have to collect to make it put in one platform that we can have easily accessible for the public or the especially for the scientists or operational way, for example, in the put and we integrate the biogeochemistry data to the existing platform like the ocean observing system that already employ the physical parameter, for example, in the operational institute like BMKG, then we can use the data more useful and, for example, to predict the extreme event like eutrophication, harmful algal bloom, or potential deoxygenation or hypoxia and stuff. Then, since the main problem is the cost of the kind of the how we integrate the biochemical variable monitoring or data collection, is that then we need to of course for the government or funding agency that will potentially give the funding for that kind of observing system that also include not only the physical aspect but also biochemical or even biological aspect, then we need to make like cost benefit analysis that will show the policymaker how much cost needed for that kind of observing system that integrate all of the variables compared to what kind of benefit and how much the benefits can be obtained for that kind of observing system. So up to now, we still in progress to provide that kind of cost benefit analysis for the integrated ocean observing system. And so hopefully within that kind of analysis, then the funding agency or especially the government that will run the system will take more attention in integrating the ocean observation that not only rely on the physical aspect but also the biological and biochemical. Thank you.
Yeah, thank you, Prof. Aan. Yes, especially for people who work in modeling, they need observation for validate the models, for example. And now in oceanography data, we have like Earth Ocean database, and hopefully in the future we will have database only for Indonesia. I hope so, not only for ocean physics but also for biogeochemistry parameter. I think that will, yeah. Okay. Is there any question for other audience? We still have time. Maybe I will come back to Dr. Bernhard Meyer because I invite some of my students here, students from Oceanography department. And could you please maybe give them some, you use models for a long time, right? You use HamStorm and EcoHam. Maybe you can give them some insight what kind of programming language did you use, and maybe give them some motivation of how to say, to learn models, maybe for them.
Yeah, the models usually in science, I think in oceanography and biogeochemistry and so on, models are written in Fortran language, which is IT people say it's a number cruncher because it's a very fast programming language which is able to process lots of data, which we need if we do modeling. So the models themselves are written in Fortran language. And besides that, because this is running on Linux computers, we use the C shell or any other shell language, actually, which is a script to put everything together. So the model is only one executable at the end after it has been compiled. And the executable then has to know where the input data come from, where to put the output data, and so on, the model results. And this is all written in a shell script. So I prefer the C shell, but other people may prefer other things, corn shell or whatever. So this is also something we need to know, and essentially also about Linux itself. So usually we are running this model system on a supercomputer in parallel mode, which means that many processors are calculating in parallel. So this works in a way that we divide the model domain into stripes, east-westward stripes, and each stripe consisting of a few model rows only is calculated, or the equations are calculated on a separate processor. And this increases the speed of the simulations enormously. So what we use for these simulations, for the HumSum, I think we used 96 processors or something like this. For the EcoHum as well, the ecosystem model. So this would be something, yeah, a little bit knowledge about this would be good to run these models.
Okay. So I think it is interesting that you mentioned that Fortran is like a number crusher or something like that, because yeah, in ocean modeling, the almost all the model in oceanography is still using Fortran as the programming language. So I hope they give my student motivation to study this kind of language. This because they are still in the second years of years, and they learn first time Fortran. So they want to know why we should learn Fortran, which is really old programming language. But in fact, in oceanography, we still use it as a really powerful
Yeah,
programming
it
language.
is. It is a kind of old language, but it's
Yeah.
still up to date because they improve it, and even C language can be included into Fortran. And so it's really, I think it's a very good language. And it's also of advantage to learn, really, to have a class, a course about Fortran language, because you can how to say, you can write a program in this way or that way, and this way may be slow when it's running, and the other way may be fast. So you need a little bit of background knowledge how data are saved, for example, in the computer system, to be able to really make a fast program, to create a fast program. Yeah.
Yeah. Thank you very much, Dr. Bernhard. And I have a question from the chat box. This question for Prof. Aan. I think I will write it. Okay. So first question is, what do you see as the most urgent research priorities for advancing marine biogeochemistry in Indonesia? This is the first question. And the second question is, what are the biggest gaps in current research capacity? For example, like instrumentation, expertise, data sharing that need to be addressed for future progress. Yeah, Prof. Aan, please.
Yeah, so we, according to the studies, is that we actually still have 5 main themes that can be used for the future research in marine biochemistry in Indonesia. So that maintain can be considered as the most urgent research priorities. Of course, for especially for the marine pollution, it's since the according to the publication we already have that already in condition of the saturated studies, which is many studies about marine pollution. But so then for the next stage is that we can use that kind of the data or the result for the integrated with policy making, especially how to manage our environment in marine environment in Indonesia. And that's also the how we will proceed with the next stage of the research. As for the other themes that are still emerging or even still lacking, then we have to make it progressive enough so that the research will move to the better condition. But of course, as you mentioned of the question number 2, that there are gaps in the current research capacity, as you already mentioned, that we also have the advancement of the instrumentation, including analysis tools, including collecting data tools and extra. And it's also we aware that is also the gap of the current research capacity. And of course, the expertise is also the another challenge. That's why we have a mission here in the IMBF to make a talent pool of the marine biogeochemistry researchers in this platform, so that we can know each other and we can collaborate in the future. So, and also those who still in the career early career path, that we can give the capacity development for so that we have more expert in the future. And of course, you mentioned about the data sharing or data exchange. As I mentioned, that we actually have a national platform to like repository. We can start with that, and if in the future we have the more open access to the kind of repository or like that we know together, for example, in the Copernicus platform in the Europe Union or extra, then we can, of course, it's the progressive movement in the future. I think that's my answer. Thank you.
Okay, thank you, Prof. Aan. We have another question from the chat box. It's this question for both Prof. Aan and Dr. Bernhard Meyer. This is the question from my student. He said, are there still major unsolved problems in the field of oceanography, maybe in general and maybe in biogeochemistry? And in your opinion, how could they be addressed? So maybe Prof. Aan first.
All right. Yeah. There are actually many things that we don't know about our ocean, especially. So that's why our research and studies still progressing. So that's why we still have the many students that study about the marine science and oceanography as well. So, for example, that what's actually needed by the public or the policy maker is that how we can actually predict when the Hamada bloom, for example, will happen, or how the how the eutrophication or like hypoxia will happen, and etc. how it's progressing and how it will affect the ecosystem. Then the capacity to predict or to forecast that kind of phenomena that's really on the oceanography parameters, not only the physical aspect, but also the biochemical or bio aspect of oceanography still needed. So, of course, the we know the challenge arise, but that's kind of the capacity to predict, to forecast is absolutely needed by the policymaker, since they dealing with how manage the ecosystem, how to manage the coastal, and how to manage the whole Indonesian Ocean. So I think our contribution not only us that already in the marine institution, but so all of the students here that learn about oceanography or marine science have to contribute to the progressing of the marine science in Indonesia.
Okay, thank you. Maybe for from Dr. Bernhard, your opinion.
Yeah, I could add that there are some, at least some, if not many, processes which are not yet fully understood. Just as an example from physical oceanography, there is still going on research, for example, about the energy transfer, from let's say from turbulent water to heat, for example. So there is energy and the turbulence is decreasing, but where does it go? Where does it stay, the energy, There's nothing, yeah, we have constant energy in the system, but how does it change from one state to the other state? So this is one thing they are doing research about, and in biogeochemistry, there is a lot still not yet fully understood. As I said, for example, the degradation of TDOC is not yet really surely understood, and we investigated this quite nicely for the Indonesian seas, for example, for the tropical seas, but maybe we have a totally different TDOC system in the somewhere else, in the tropical sea, for example. So there are many dependencies, and another example would be the process of calcification is also, as far as I know, not yet really fully understood of corals. How do they really produce aragonite? Aragonite is a certain type of CaCO3. It's the internal frame of corals. How does it really happen? And this is still also under investigation. So there are many, many other examples of things still not known in this field. Thank you.
Thank you very much, Prof. And Dr. Bernhard Meyer. I hope that answer your question. Yeah, the name or Raja, that is the name of my student. Okay, is there any question? Maybe last question from the audience before I close this session. Let's see. Okay. Okay. I think there's no more question from the audience. Maybe last few comments from Prof. Aan or Dr. Bernhard Meyer.
Yeah, for the student, it's keep your spirit, and yeah, we as the one of the challenge in the marine biodiversity that is Indonesia is expert, the availability of the expert. So it's the moment that we do the capacity building for the young generation of the scientists in Indonesia. So it's also my pleasure to see that students can join this kind of webinar so that we can do more in the future. Thank you.
Thank you, Prof. Aan. For Dr. Bernhard, any comment?
For my, I just can also say to everyone actually, but particularly to students as well, stay curious. Always ask questions. That's all. Thank you.
Thank you very much. It's really great to have Prof. Aan and also Dr. Bernhard to give us a really interesting presentation. I hope it gives us a really fruitful insight and also for future potential for future research. And thank you very much for all participants for joining us this afternoon. And we look forward for the next webinar, I hope next month in October. Thank you very much once again, and good afternoon. I close this session. Thank you.
Thank you.
Thank you. Thank you,
Thank
Bernhard,
you,
for training
Bernhard.
us.
Thank you also to you, and congratulations to your professor title.
Thank you very much.
Okay, bye-bye.
Bye-bye.
Bye. Thank you very much. Oke. Mbak Ida, ini boleh saya sudah di close ya?
Boleh, Mbak, boleh.